Today's medical progress has enabled prevention and treatment of many diseases, even previously fatal ones. Our ability to intervene and mitigate disease progression depends on our understanding of their various mechanisms. Fortunately, we can realize this goal through research on model organisms and then apply our knowledge to humans. České Budějovice is a charming South Bohemian town and the seat of the Biology Centre of the Academy of Sciences. Scientists of the centre use in their research various model organisms, in particular the tiny fruit fly Drosophila. Why? It's because its genetic information is surprisingly similar to our own, and it can be easily manipulated for research purposes. The advancement of scientific knowledge leads us directly into the fruit fly egg, containing all the genetic information responsible for the control of fruit fly development, appearance, and lifespan. We are able to manipulate genetic information stored in the DNA molecule. The results of our intervention are often destructive rather than creative. Usually we select an interesting gene, for example based on a similarity with a human disease-associated gene, and scientists investigate the role of such individual genes by disabling them and observing what happens. The genes are deliberately disabled using molecular scissors, so-called talons, that remove specific parts of DNA molecules. Cells try to repair such damage, but as a consequence, some errors usually remain. Switching off the gene has a consequence, a so-called mutation. There can be many mutations, each with different manifestations. It is not always easy to determine what the consequence of the loss of a particular gene will be. Fruit flies can have easily detectable tumors, similar to those observed in humans. By switching off various genes, scientists can identify which genes normally act to suppress tumor progression. Fruit flies and the characteristic changes in their properties caused by mutations are examined in detail with the help of unique instruments. For example, some tests include measuring changes in mobility. The flight simulator allows the monitoring of flight parameters in responses to changes in the height of horizon, fatigue, and other factors. The research is also focused on respiration. This instrument measures oxygen consumption of 8 individual fruit flies simultaneously. Also, the Drosophila heart, that is located on the dorsal side of the body, is investigated in detail. The fruit fly heart beats up to 4 times per second. Investigations can be accelerated by performing experiments using tissue cultures. Such work, and especially the interpretation of the results, are easier than in studies with whole organisms. All this research pursues a single goal: to uncover the mechanisms responsible for the formation of tumors, cardiac, neural, and other diseases in humans, to give hope to millions of people around the world who suffer from and are killed by such diseases every year. The evolution of life on our planet has been challenged with periodical changes in light and darkness, and with seasonal weather oscillations. It is therefore not surprising that living organisms adapted well to these environmental cycles. Animals and plants are not surprised by everyday sunsets or by change of season. They can predict such events and prepare for them in advance. Their responses to environmental cues are imprinted in their genetic information. Virtually all species on Earth have their own endogenous timekeeping mechanism. Such mechanisms are referred to as their biological clock or seasonal calendar, depending whether they measure diurnal or seasonal time. Disruption of the human circadian clock can result in serious health problems, ranging from sleep disorders to higher incidences of cancer. Studies of insect circadian clocks enable the scientists in České Budějovice to understand how the clock functions in humans. They've discovered that evolution has found different solutions to the same problem. For instance, the genetic clockwork of the linden bug resembles more the human clock rather than that of the fruit fly. Therefore, suitable insect model species help us to unravel the functioning of the human biological clock. Scientists also endeavour to understand how the biological clock participates in the measuring of seasonal time, characterised by changes in the duration of day and night. The seasonal calendar governs the entire life cycle of plants and animals, including insects. It determines the time of reproduction, changes of generations, and also the timing of the dormant state for overwintering. Insects are the most widespread and numerous group of organisms on Earth. Many species transmit serious diseases, others represent significant agricultural pests. Understanding their seasonal calendar will help to facilitate the regulation of their populations. During overwintering in a dormant state, insects attain very high tolerance to cold and frost. Most species avoid freezing by suppressing the formation of ice crystals at body temperatures as low as -50 degrees Celsius. Other species, on the contrary, trigger ice formation in their bodies and survive in a partially frozen state. Some insects can be stored and remain viable for almost infinite time at the temperature of liquid nitrogen, that is, -196 degrees Celsius. Understanding these processes in insects can help solve the problem of long-term storage of other biological materials, for instance, human cells and tissues. The Czech Republic belongs to those fortunate spots on the Earth that are not threatened by malaria and other diseases transmitted by blood-sucking arthropods. Only ticks transmitting the encephalitis virus and Lyme disease present a serious menace. We can protect ourselves against tick-borne encephalitis by vaccination. However, an efficient vaccine against Lyme disease spirochetes still does not exist. How does the tick life cycle look? Six-leg larvae that hatch from eggs feed on small animals, such as rodents. After feeding, they drop off from the host and during the next 6 to 8 weeks molt to the 8-leg nymphs that can attack larger hosts, including man. The nymph is the most dangerous stage in terms of Lyme disease transmission risk. The nymph feeds for only 3 to 5 days and, owing to its small size, is often overlooked. Fertilized adult female ticks feed on their hosts for about 1 week. After dropping off, each female can lay between 1,000 and 2,000 eggs before dying. In order to develop efficient tick control strategies and prevent the transmission of pathogens they can carry, we have to understand the interactions taking place at the tick-pathogen interface. The discovery of a two-prong vaccine that would protect against tick infestation and thereby prevent transmission of tick-borne diseases is the desired research goal. Current research is focused on tick saliva that contains compounds suppressing pain and inflammation. This is why the feeding tick may remain unnoticed by the host. Other components of the saliva restrain the host's immune response and thereby facilitate pathogen transmission. Another research theme relates to concealed antigens, mainly proteins from the tick gut that are important for pathogen persistence inside the tick. It is anticipated that vaccination with either type of antigen should elicit the production of antibodies that will block vital physiological processes in the attacking tick. The role of specific tick molecules during pathogen transmission can be elegantly studied using the RNA interference method that makes it possible to specifically switch off the synthesis of individual proteins. The lengthy path to efficient protection against ticks and, for instance, to the development of a potent vaccine against Lyme disease will be paved by the acquisition of a complex mosaic of reliable pieces of knowledge. No shortcuts are possible. Investigations on model species are also important in plant research. The rice plant serves as the preferred model for the monocot species and the cress plant Arabidopsis thaliana for the dicotyledons. This plant is a small annual weed that can be found in gardens, fields and rubble. For plant geneticists, Arabidopsis offers great advantages. It has a short life cycle. Seeds from a new generation can be obtained in just a few weeks. The fact that it has a small genome, in fact the smallest in the plant kingdom, is even more important. The entirety of the genetic information is packed into a relatively short length of DNA. The number of genes is comparable to other plants or even to humans, but there is a significantly lower proportion of non-coding DNA. These DNA properties make Arabidopsis more accessible for mutagenesis. The Department of Molecular Genetics at the Institute of Plant Molecular Biology takes advantage of advanced Arabidopsis functional genomics in its research of the hop plant. Hops are not only the raw material for beer production, but also an important source of pharmaceutically valuable compounds that have anti-cancer and anti-inflammatory effects or mimic the effects of some human hormones. The best known of these substances is called xanthohumol. The biosynthetic pathway yielding xanthohumol is similar to the Arabidopsis pathway that generates dyes for seed coat coloring. If we introduce a hop gene, whose function we only anticipate, into Arabidopsis, we can confirm or exclude our prediction. In Arabidopsis research, there exists a large collection of mutants. Every gene has been mutated at least once. To know more about the function of a gene of interest, we can test the ability of a hop gene to functionally replace the mutated gene of Arabidopsis. In other words, we can find out if the function of hop gene is similar to that of the well-known Arabidopsis gene. For example, we have successfully recovered trichome production on the leaves of Arabidopsis hairless mutant using a hop gene. With this approach, we can also examine consequences of the overproduction of a hop-specific protein in Arabidopsis. The reciprocal approach is also possible. Arabidopsis genes can be overexpressed in hops. In collaboration with Professor Weber of Hohenheim University in Germany, we have managed to transfer an Arabidopsis gene into the hop genome. The result was a plant with beautiful red cones. Although such a change has no economic value, this proof of principle opens the door for introducing other, more relevant genes into hops. Investigations performed with Arabidopsis and other model organisms do not bring immediate profit and may seem purposeless. However, research focused on knotty problems is a long-term investment. For example, several genes implicated in tumor development had been discovered in the fruit fly many years before this information was exploited in medicine. The results of current research will eventually influence the treatment of diseases, lead to development of vaccines preventing tick-borne diseases, and improve the recovery of beneficial compounds from plants.